We will look at blood cell counters in this video, which are also known as hematology analyzers. In this video, we will look into the beginning of blood cell counters, the working principles of blood cell counters, types of blood cell counters, and components of blood cell counters. Let's begin with the beginning of blood cell counters. Before automated cell counters were introduced, complete blood count tests were performed manually. White and red blood cells and platelets were counted using microscopes. The first person to publish microscopic observations of blood cells was Antoine van Leeuwenhoek, who reported on the appearance of red cells in a 1674 letter to the Proceedings of the Royal Society in London. In the late 1940s, Wallace H. Coulter, motivated by a need for better red blood cell counting methods following the bombing of Hiroshima and Nagasaki, attempted to improve on photoelectric cell counting techniques. His research was aided by his brother, Joseph R. Coulter, in a basement laboratory in Chicago. Their results using photoelectric methods were disappointing, and in 1948, after reading a paper relating the conductivity of blood to its red blood cell concentration, Wallace devised the Coulter principle, the theory that a cell suspended in a conductive medium generates a drop in current proportional to its size as it passes through an aperture. Let's look at the principle of the blood cell counter. While under contract to the United States Navy in the late 1940s, Wallace H. Coulter developed a technology for counting and sizing particles using impedance measurements. The technology was principally developed to count blood cells quickly by measuring the changes in electrical conductance as cells suspended in a conductive fluid pass through a small orifice. Presently, over 98% of automated cell counters incorporate this technology, which is referred to as the Coulter principle. In the past 75 years, the technology has also been used to characterize thousands of different industrial particulate materials. In a Coulter counter instrument, a tube with a small aperture on the wall is immersed into a beaker that contains particles suspended in a low-concentration electrolyte. Two electrodes, one inside the aperture tube and one outside the tube but inside the beaker, are placed and a current path is provided by the electrolyte when an electric field is applied. The impedance between the electrodes is then measured. The aperture creates what is called a sensing zone. Particles in low concentration suspended in the electrolyte can be counted by passing them through the aperture. As a particle passes through the aperture, a volume of electrolyte equivalent to the immersed volume of the particle is displaced from the sensing zone. This causes a short-term change in the impedance across the aperture. This change can be measured as a voltage pulse or a current pulse. The pulse height is proportional to the volume of the sensed particle. If constant particle density is assumed, the pulse height is also proportional to the particle mass. This technology is also referred to as aperture technology. Let's look at the types of blood cell counters. There are mainly two types of blood cell counters that are present in the technology. One is a 3-part cell counter, and the other is a 5-part cell counter. First, we'll look at the 3-part cell counter. A 3-part differential cell counter uses Coulter's principle to find the size and volume of the cell. The sample is lysed and dissolved into an electrolyte solution in a container, which also holds a smaller container. The smaller container has two pumps running to and from its solution, one creating a vacuum and the other replacing the lost solution. The smaller container has a small hole, an orifice, near the bottom of the container. Coulter's principle is applied through the use of two electrodes. One electrode, the internal electrode, is within the smaller container, and the other, the external electrode, is outside of the smaller container, but within the electrolyte sample solution. As the vacuum draws the sample cells through the orifice, the cell momentarily causes electrical resistance to the current as it passes through the orifice. This resistance is recorded, measured, amplified, and processed, which can be then interpreted by the computer into a histogram. The 3-part analyzer is able to differentiate between 3 types of WBCs: neutrophils, lymphocytes, and monocytes. The second type of blood cell counter machine is a 5-part hematology analyzer. This type of hematology analyzer utilizes both Coulter's principle and flow cytometry to determine the granularity, diameter, and inner complexity of the cells. Using hydrodynamic focusing, the cells are sent through an aperture one cell at a time. During this, a laser is directed at them, and the scattered light is measured at multiple angles. The absorbance is also recorded. The cell can be identified based on the intensity of the scattered light and the level of absorbance. A 5-part cell counter can differentiate all WBC types: neutrophils, lymphocytes, basophils, eosinophils, and monocytes. 5-part analyzers are more expensive than 3-part analyzers, but provide much more in-depth information about the sample. Specific jobs, such as allergy testing, require 5-part differential analysis. However, most medical tasks can be completed with the 3-part analyzer. Let's end the video with components of blood cell counters. There are 4 major components of blood cell counter machines: fluorescent flow cytometer, electrical impedance, flow cytometers, and reagents attached to the machine. In fluorescence flow cytometry, or FFC, it is used to analyze physiological and chemical properties of cells. It can also be used to analyze other biological particles in urinalysis analyzers. It provides information about cell size and structure and information about a cell's interior. Flow cytometry is a technique used to detect and measure physical and chemical characteristics of a population of cells or particles. In this process, a sample containing cells or particles is suspended in a fluid and injected into the flow cytometer instrument. The sample is focused to ideally flow one cell at a time through a laser beam, where the light scattered is characteristic to the cells and their components. Cells are often labeled with fluorescent markers so light is absorbed and then emitted in a band of wavelengths. Tens of thousands of cells can be quickly examined and the data gathered are processed by a computer. The reagents used in hematology analyzers are cleaners, which are used to clean bath or chambers in the measuring unit. Diluent, which is normal saline used for the dilution of blood samples to the propionate for the cell counts. Hemolysius, used in vitro quantitative detection of white blood cells, number in the whole blood and hemoglobin concentration, etc. As usual, we try to keep this video for simple understanding of hematology analyzer machines. Hematology is very wide and deep in study, so if you would like to